Hello friends. So already we have made the 43S pre-initiation complex. Now we have to make the 48S closed complex that will change into 48S open complex. So for these two complexes to form, first our mRNA has to combine with pre-initiation complex. So for that to occur, first let's see what happens to our mRNA. In previous videos, we have seen the structure of mRNA. It has got a cap at 3' end and it has got a poly A tail at 3' end. And as it's a single stranded structure, so many times it gets folded upon itself to form many hairpin loops. These are the hairpin loops like this here, here, here and here. There can be many folded upon itself structures or the hairpin loops. So what happens? First, initiation factor F comes. This initiation factor F actually is the combination of many initiation factors like 4G, 4A and 4E. This 4E factor or the 4E initiation factor actually combines with cap of the mRNA here on one side, and on the other side it combines with poly A binding proteins here. This is the poly A tail. There are many proteins or the poly A binding proteins attached to this tail. This 4E combines with these proteins on one side and cap of mRNA on the other side, so that this mRNA actually gets folded upon itself. Similarly, 4G also can combine with poly A binding proteins as well as with 4E. So, how the 4F is formed? 4F is actually a combination of 4E here and 4G here and 4A. All three of these have got different functions. Function of 4E is that it gets the mRNA molecule to get folded upon itself because it's binding with the cap on one side and PAB or poly A binding proteins on the other side. Similarly, 4G is also bound to the poly A binding proteins on one side, and this 4G actually guides mRNA to 43S pre-initiation complex. The function of 4A or 4B is that it has got a ATP dependent helicase activity. This activity uses ATP or energy and it removes the hairpin loops here. What happens? mRNA in eukaryotic cells gets bound to 4E, which makes it to folded upon itself, and 4G which guides it to pre-initiation complex, and 4A which actually uses energy and removes the hairpin loops here. So this is the structure which gets bound to the pre-initiation complex. Now here is our start codon here, or AUG, which is the start codon. This codon actually is surrounded by a Kozak sequence. Kozak sequence is the consensus sequence which is present in the eukaryotic cell. So let's see what is the Kozak sequence actually. Here is our AUG, that is adenine at +1 site, then U at +2, then G at +3. This is AUG in Kozak sequence. After that, one more G is present. So here actually there is one more G actually in mRNA, and before that is GCC or GCC. That is, before adenine there is cytosine, before that is cytosine, before that is guanine, Then this is P4, that is at -4 position there is any purine. Before that is C, then G. So what is the Kozak sequence? It is actually GCC, purine, GCC, adenine, UGG. So this is the Kozak sequence which actually incorporates start codon in its structure. So now we have got this structure, that is mRNA, which is folded upon itself and is bound to poly A binding proteins and initiation factor 4E, 4G, 4A and 4B also here. The cap is bound to the tail with the help of 4E, and we have got our start codon here, which is incorporated in the structure of Kozak sequence in case of eukaryotes. So now we have formed 43S pre-initiation complex on one side, and on the other side we have seen how the initiation factor 4F gets attached with the mRNA and folds it upon itself. First 48S closed complex is formed. For it to form, our mRNA, which is attached with the initiation factor F, which we have seen is the combination of 4E, 4G and 4A. So in the whole it can be written as initiation factor 4F. So mRNA which was bound to the initiation factor 4F, that comes and gets bound to the 40S subunit of the ribosomes. We have seen in the previous slide also that actually 4G subunit of 4F guides this mRNA to the 40S subunit. So for the sake of simplicity, we have shown it as a straight line. This is cap 5' cap, which is attached to the 4F, and this is our 3' tail. Here are many adenine moieties and many poly binding proteins also. This actually is folded upon itself, but for simplicity we have shown that this is our 40S subunit, this is the P site, this is our ternary complex which is bound to the P site. These are our initiation factors 5, 3, 1 and 1A. This structure was actually part of 43S pre-initiation complex. Now our mRNA, which is attached with initiation factor F, comes here and gets attached with the 40S subunit. So this complex is known as 48S closed complex. After that, ATP dependent scanning occurs to locate the start codon. So our ribosome or the 40S subunit of the ribosome moves upon this mRNA so that the anticodons on this transfer RNA actually gets bound to the start codon in the structure of mRNA. So when they gets bound, this whole structure is said to be transferred into a 48S open complex. So 48S open complex is formed when 40S ribosomal subunit, which was moving upon the mRNA, gets to locate the start codon, and a bond is formed in between the AUG start codon and the corresponding anticodon in the initiator tRNA. So now this structure is called 48S open complex.